We are moving on to deuterostome animals. Okay, so here are the objectives that you can read over. So deuterostome animals, so these are animals that during embryonic development, during, more specifically gastrulation, they form the mouth second. So if you remember from the protostome uh, PowerPoint, uh, deuterostome means second mouth because deutero means second, stoma means mouth. So these animals form their mouth second. Okay? And the coelom develops from outpockets of the mesoderm. Okay? But also, if you recall from the protostome PowerPoint, that there is, so this was the traditional thought, okay, that deuterostomes develop the mouth second, anus first, and protostomes develop the mouth first and the anus second. But there's a lot of just as they studied more animals and the way that they developed, they found that in protostomes, there's really a lot of variety because some protostomes do develop the mouth first and then the anus. Others develop the mouth and the anus at the same time, and then others develop the the anus first and then the mouth. Okay, so there's just a lot of variety with protostomes. But here you can see a phylogeny of deuterostomes because this is based on, phylogenetics and also molecular data. So in deuterostomes, you can see that it is not as diverse as protostomes. So every animal that we've looked at so far has been a protostome. Okay, so like the flatworms, the ringed worms, um, mollusks are all, and arthropods, they're all protostomes. Okay. The deuterostomes includes Echinodermata, Hemichordata, and Chordata. So these are the phyla. Echinoderms include things like sea stars and sea urchins, uh, also like sea cucumbers, sand dollars. Hemichordates, these include things like acorn worms, which are here on the left. So these are animals with worm-like bodies that either deposit feed or suspension feed. And also the pterobranchs, which can also, suspension feed. Okay. And then for the phylum Chordata, this includes the torpedo-shaped lancelets, okay, like this animal here, kind of looks like a fish almost. And also like the sac-like tunicates, so these animals here. And then the other group are the vertebrates, so animals with backbones. So Chordata would be the phylum here, and then Vertebrata would be the subphylum. So for example, humans, we're from the class Mammalia, okay, which is from the subphylum Vertebrata and the phylum Chordata. Invertebrates are just animals without the backbone. Okay, so about 95% of the known animal species are invertebrates. But when you look at deuterostomes, the vertebrates are the ones that are dominant here. So deuterostomes have a lot of diversity. They have a lot of diversity in the body plans, the feeding methods, the way that they uh, move around, and also how they reproduce. But we'll start with the echinoderms. So echinoderms, these are the uh, animals that are named for the spikes that they have on their body. Okay. spiky skin. That's what the name translates to. Okay. And echinoderms are all marine animals. And to date, about 7,000 species of echinoderms have been described. And they're very abundant, especially in deep water environments. But you can also see them uh, in shallow water, like intertidal zones. So you'll be able to see things like sea urchins or sea stars. The three main traits that identify echinoderms as a monophyletic group are radial symmetry in adults. Okay? And I say adults because, as in the larval form, they do have bilateral symmetry. The second trait is they have an endoskeleton of calcium carbonate, and they also have a water vascular system and tube feet. Okay, so they do have the radial body symmetry as adults. The larvae have bilateral symmetry, okay, and the as similarly to the cnidarians, which are also radially symmetric, these animals do not have heads. Okay, so they do not have heads. They only have a mouth, so that's called the oral end, and then the opposite side of that would be the aboral end. So when you look at a sea star, you're looking at the dorsal side of it. The mouth would be on the ventral side underneath. Okay, so that would be the oral end. The top part without the mouth, that would be the aboral end. Okay. So the adults have they do have radial body symmetry, and because the the number of planes that it divides into is five, it's also called penta radial symmetry. And this is and the penta radial body symmetry originated early in echinoderm evolution. So here you can see an echinoderm larva or larvae, okay, and they do have bilateral body symmetry. So there's only the body divides. If you divide the body down the middle, it only has one plane of symmetry. Okay, so one side would be the mirror image of the other. But in adults, they have radial symmetry, so multiple planes of symmetry. Like this is a sea urchin, okay? But it's just the skeleton of the sea urchin. It's missing the spikes. So echinoderms do have an endoskeleton, just like you can see here with the sea urchin. That's made of calcium carbonate. Okay, it's a hard, protective, and supportive structure that's located underneath the thin epidermal tissue. Okay, you can see also in sea stars, it's right below the skin of the sea star. So all of this is the endoskeleton. Okay. The endoskeleton also has what's called catch cartilage, which allows it to kind of lock into place. So that's why when you see sea stars, usually in intertidal zones, they don't move because of that catch collagen. So they're able to lock their bodies into place. Because even though the skeleton is sturdy, this endoskeleton, it is also flexible. Okay, so the echinoderms, they are also defined by the water vascular system. So this is a system that they use for locomotion and also for feeding. So it's a series of branching fluid-filled tubes and chambers, and the water flows in and out of this system. So the water, so this is the top of a sea star that you're looking at here. And at the very top of the sea star on the aboral end, you can see it's like a sieve structure here where the water goes in. So the water would go in through this structure here, then it moves into these various canals. So there's like this is called the Stone Canal. There's the Ring Canal that leads into the radial canals, and then eventually it goes into these smaller structures that are called the ampulla. And the ampulla are located on the inside of the animal, but those lead to the tube feet on the outside of the animal. So you can see the 2 feet sticking out from the bottom here. So that's what they're using for locomotion. Okay? They're using a hydrostatic skeleton on the in the 2 feet. Here you could get a better look. Okay? So the 2 feet on the outside, the ampulla would be on the inside. Echinoderms use most methods of feeding, so they can either use the mass feeding when they feed on things like algae or other animals. They can use suspension feeding, and they can also use deposit feeding. The mass feeding, this would be something that you would see with sea urchins when they graze on algae, or when you see sea stars that prey on other animals like bivalves, which are clams, oysters, things like that. Suspension feeding, you can see that with the water lilies, and deposit feeding, you can see that with things like sea cucumbers. Okay. So remember that deposit feeding is when they animals take in soil or some other substrate, and along with the soil, they would take in uh, organic matter. But the 2 feet, so in addition to playing a role in locomotion, the 2 feet also play a role in obtaining food. So this is true for things like sea stars because they use the 2 feet to open bivalves. Because what they do is they grab onto the bivalve, they'll crawl over the bivalves, they'll lock into position, and then they're able to open the shells or split the two valves apart, and then they'll evert their stomach into the bivalve, and then the stomach will produce the enzymes, and when it breaks down the bivalve tissue, it'll just absorb the nutrients. Okay, so you can see things like um, how sea stars are important predatory animals. So like the Pisaster ochreus, so this is a type of sea star that when removed from study plots, they did find that these animals are considered to be keystone species because they affect other animals in those communities. So in the 1960s, Robert Payne removed sea stars from half of his study plots, okay, and the other half were control plots where he didn't remove the sea stars. And he saw that the number of species in the communities where he removed the sea stars plummeted. Okay. So where before you would have, a higher diversity of, algae and other animals, other invertebrates, when you remove the sea stars, those communities just became overgrown with strands of mussels because that's what the sea star eats, or that's the main thing that they prey on. Okay? Things like sea urchins are herbivores. So sea urchins, they have a jaw-like structure in their mouths that's called Aristotle's lantern. Okay, and it's also made of calcium carbonate, and they use um, this tooth that can be extended and retracted during feeding. Sea urchins are also important grazers. Okay, and another something else that they found was that when you remove the predators of the sea urchins, they can that can also affect communities because in this case, the sea otters would be the keystone species, and when the keystone species are removed, then the animals that they prey on, there's nothing to control those populations, so those populations explode. And when you remove the sea otters, then the sea urchins uh, they overfeed on kelp forests, so they can destroy the kelp forest. And here are the five major lineages of echinoderms that are living today. So you have the Crinoidea, which are the feather stars and sea lilies. You have Asteroidea, sea stars. Ophiroidea, these are brittle stars. Yeah. Echinoidea, sea urchins and sand dollars, and then the Holothuriidea, which are the sea cucumbers. So here you can see a water lily or sea lily, and this is an example of a sea cucumber. Okay. Okay, so we'll pause here and then we'll continue on the next one.